pokrenut, ako budeš mogla pokrenuti, to je to onda, znaš, nije problema. Na 4 minute. Okay. So let's continue. Imamo kjer priča na engleskom, lakše mi je nego na naše nekako, kad zbog pogotovo kad nisam koncentrisan, da ne znam što pričam. So I'll just continue in English. And today we're actually going to just do a quick recap of the stuff we did last week and try just quickly to refresh our memory on um, what we have said, what we need in order to do some analysis. And one of you sent me an email that he or she couldn't download um, some of the stuff we were downloading via those links and um, those kind of publicly available data. That's perfectly fine because I think those I forgot completely that you do not have computers with roughly a terabyte of hard drive. So downloading all this stuff would take up a lot of your computer space. So what I will do in the following weeks and days, I'll make those files a little bit more accessible to you by choosing data files from only one chromosome. We can then, instead of doing the whole 23 I mean, it's the same thing, right? So once you know how to analyze one, you will be able to analyze all 23. It doesn't really matter. So I'll chop up data files into one or two or three chromosomes, including the reference data to it, and then we'll be able to uh, I'll put it on in a Google Drive thing, and then you'll download it from there, because I think it's just more applicable for you, for your computers, to be able to have enough space to run other stuff for daily activities in life, right? So, how many of you have previously been using R for anything, for visualization, or just how many of you have heard about it as a software tool?
I heard about it and I did some basic things, very beginner stuff.
Okay, anybody else?
Not really.
Okay, not really. How many of you have managed to install uh, R on your computers? Somebody's raising your hand. Just speak. Oh yeah, that's answering the question, right? Okay.
Yeah, I just wanted to say that I've installed it.
No, yeah, that's the smart way of answering. Great. I'm an idiot. I didn't figure it out. Yeah, makes perfect sense. So yeah, so I see. Let's say uh, half of you kind of installed it, right? But let me see who is here, who is not here, because unfortunately I have to put this attendance stuff. I always forget. I forgot also last time, so I just put it. Everybody was there. They're actually really insisting on this stuff. Let me see attendance. Okay. So I'll again just call up your names and then uh, say there or not. Adna,
I'm here.
Aisha, Aisha, no. Armin,
Here.
someone is writing. Okay, she's here. Berina,
Here.
Igor,
Two.
Ilham, Ilham, you too, yeah?
She's on her way back home from the university.
Okay. Irnes,
Here.
Yasmina,
Here.
Lamia, Lamia joined. Leila, Leila nije tu, jel? Dobro. Nerma,
Here.
Sara,
Here.
Semina,
Here.
Tarin,
Here.
Đorđe. Dobro.
I'm here.
Dobro. Okay. Message. And, okay. So several of you are able and were able to install R, and we'll now have a look just into a quick setup on what we need in R in order for us to do some basic analysis with genetic data, right? So let me share my screen, entire screen share. Good, can you see my R interface on my screen?
Yes.
Okay, good. So this, you do not, this is something else, this is for my work, but when you open R, basically you'll be, once you install it and all the stuff, right, you need to install first R, the software, and then R Studio, this kind of visual interface, right? Once you open it, it's going to be a little bit not in this order, the script where you can write it, the console, the terminal, the background, and this package thing. So I would suggest you to just quickly Google or YouTube, actually it's much quicker, kind of R layouts, and you will have here in view a lot of options how to make it the most sensible for your way of working, right? I mean, I think maybe it would be the best to make a layout similar to mine, so you can follow the easiest, because I usually type here in the script and then execute it from here rather than using the console here, which is typing. So basically here you can type and edit and press another line, like writing the whole script, while here you execute commands by pressing enter, and here you, in order to execute a command, need to hold shift, control and enter, but also you can make like hashtags where you just write some explanation on what you did, right? You see, when you write normal text, it could be a command and it's normal letters, but if you put the hashtag, it doesn't read it and it's not calling it up, basically meaning that you can write as much as you want to comment. So if you do something, then you can write yourself, we did this because of this and this and this, and it can all be in your script. So once you open it, you should know what's going on and what you did last time. And just something like a sheet for yourself to know what you did last time. So usually there's a way of finding libraries, and libraries is basically extra packages in this software, because R is not only for kind of the genetic stuff. You can do anything in R, basically. It's like programming language. But there's special packages which are made for bioinformatic analysis, DNA sequencing, microarray data, and such. And in order to be able to use those commands from those packages, you need to install those packages, right? And this is basically done in this way. This is a click-through kind of approach, where you go to packages and then you keep the repository here. You alternatively can download something and then use it up from here, but this is from the online depository, and you can then type packages here, right? And the first package you would need is ggplot2, right? And then you would click on ggplot2 and click like this, install. And you see, I mean, I think I have it already, but that doesn't hurt to install again. And usually the command which is used for that is install.package and then brackets and use the quotation marks to type the particular name of your package. Another important package which we need is QQman or QQman, I don't know. Sounds a little bit odd in our language, but it's kind of okay. I guess it's QQman. And then you click this. And those two so ggplot would be, as the name suggests, a plotting tool which, independent of data input, gives you nice and interesting looking plots, right? And then you would have QQman, which is a particular software package for visualizing genetic data. And I'll show you how it looks, because basically you can just Google QQman or QQman in R, and then open the package, and I'll bring it now to this screen, and then see how it looks like. This is the package made by this guy. Thank you, Stephen D. Turner. And this is the data stuff we will have. So this is all, plotting is done when you already generated and analyzed data, right? It's just a visualization of data which is already analyzed. So it's not a tool for analysis here which we use, it is a visualization. And this is kind of the last part of the pipeline of the software tools kits we used. Oh, sorry. So the what stuff we installed previously were tools which we're going to use for quality control first and foremost, then data analysis, and this R would mostly be used for data visualization, right? But usually you technically can do any kind of analysis in any tool if you know how to use the programming language. But again, I'm not a software developer or a programmer. I have no idea how any of this actually works. So I only know how to use the stuff I was taught, and that's what I'm teaching you right now. There's different ways of looking at the same problem, but once you understand how data is structured, basically if you have the know-how, it's easy to solve those problems in any, you can use Python. If you like Python, you can use R, basically anything. And what in general this software package in R does, it takes, here you can see the base pair position, right, which is here made to be 1, and we see the chromosome, which is also 1, and then we have the SNP, which is, SNP IDs are RS1 to whatever, 50,000. They're not chronologically ordered, but they start with an RS. And the P is denoted the p-value, which we talked about yesterday, and then this software takes those 4 data points, p-value, base pair position, chromosome, and RSID, so the name of the SNP or the polymorphism, and gives you a plot like this, right? So now when you look at this plot, can anybody tell me what does this plot shows us, knowing what I told you now? It's only 4 data points. What's shown on this plot?
Chromosomes on X.
Yes. What's on the Y?
It's minus log of the p-value, I think.
Exactly, right. And what would be the red line here?
Some threshold that.
Exactly, but what's the threshold? We know the threshold here.
Well, for genetic data, it should be 5 times 10 to the power of 8.
Exactly,
Yeah.
That's this line here representing, and this is another threshold which you can use for a different correction mode, or the so-called suggested threshold, right? Because just to give you, and you can mute your microphone, please. Thank you. It's just to give you an idea about, have you heard in any of your previous classes the term missing heritability? No. Okay. So basically a kind of very simple concept, right? So if you do, so let's start from the beginning and then maybe to be clearer. So until the human genome was sequenced, in what year? You should know that. When was the human sequence, complete human sequence, genome sequenced? It's the year, Sorry?
2003.
Yeah, so between 2001 and 2003. So remember when you were born, I guess most of you are around that age. That's when the human genome was sequenced. And it cost like I don't know, $2-3 billion. It was very ridiculous expensive. Now the same tasks cost $100. So you see in your lifetime how this stuff changes. But that's not the point. The point here being is that I forgot what the point was. Just what I was talking about. Um, threshold. Yes, right. This is just a threshold. So and missing heritability. So and they thought after that that they will first and foremost we thought that humans have much more genes than we turned out to have. So we only have around 20,000 genes. It was thought you're going to have a lot of genes, that every gene is one protein because you have several thousand more proteins and such. By order of magnitude, actually, more proteins in our body, and some of them we know how they look and what they do. But only 20,000 genes. And then some people figured, okay, maybe we can see how well those diseases correlate with the genetic predisposition of people. And then they calculated, those fancy statisticians, that using this cutoff thresholds, we will be basically the best way off to excluding as many false positives as possible, but also including as many false negatives as possible. And there is, with those calculations, we say that even if you would have 1 million people genotyped for the whole genome, let's say a whole genome sequencing for one disease, you would be only able to find around 80% of the heritability of disease explained by genetics. So you would have 20% of something which you know is inherited, right? Because it's obvious from the inheritance patterns, but we do not know where this inheritance comes from and where does it lie, right? And this is the term missing heritability. And great idea is actually that a lot of the missing heritability would be somewhere here around this region, something which we would exclude as being statistically significant by any means because the signal is not strong, but nevertheless it could be well that biological determinants here is much more important than the p-value. So that would be something which is kind of could be coined to missing heritability. Anyhow, with that said, this plot then represents those two thresholds, p-value on the log minus 10 scale or minus log 10 scale, in order to have this nice visualization plots, and then all the 23 chromosomes here in the line. And we only use 4 data points, which we will create using the other softwares, right? So to think about maybe in this way, you will be using this Ubuntu stuff. I mean, if we come to that, right? But let's say you will use this to have raw data, quality controlled, and then variant called for SNPs, individual positions of polymorphisms in the human genome. And then this is the software BWA, if you remember, and all the stuff which we should have installed by now. And then we will use the other software which we downloaded, Plink, to analyze those changes and get the p-values and odds ratios, which we talked yesterday about. And then once we get that output, we go with that output to R, and then in R we get the data visualization plot. It's kind of a very crude workflow, but I think it describes well what's the main purpose of genomics, right? So you go from raw data through analysis from QC to analysis and then visualization. As I mentioned, alternatively, one could use R for everything from finish to end, but this is not something I know how to do, so we will do it this way. Okay. So besides the package, so we have ggplot, we have Kukuman, and we would need also tidyverse. and for me it's all installed. You'll just follow the tutorial and get it back in, or install it like this. And then obviously, like in every programming language, you need to work with data to put some data in, and I'll just show you how it's done. This data is nothing which you will be needed to do in this capacity. It is just uploading any file from anywhere to here, and it's quite simple. And I'm showing this for you to be able to repeat it by yourself. So you go to import dataset from text base, and then like in any other click-through software, you would go and find a file which should be a text file, right? That's why it's called, sorry if I'm blowing into this stuff. Should be a text file, right, of any kind. I think also you can upload Excel sheets, but it should have either be a TXT file or CSV file, or this tab delimited, comma delimited, and semicolon delimited, or files which are that in nature but have different extensions, like in this case map or pet files. And you can see here when we upload, for example, this I think it was map file. This is the name of the file which we uploaded, right? And then we have, it says it's separated by tab, and it gives you the input file and data frame which you will observe, which you will get into R, right? And this 3, because you maybe remember from some coding classes that all those files rather need the headers in their columns, so it's easier for them to find. So if there is no headers assigned by you, right, then it assigns itself headers v1, v2, v3, v4, and if you say yes, it will technically take our first line and make it, you see, 1000 here, and it would make it XRS, blah, 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 would make it our heading, because in order for this stuff to understand what's going on, it needs a header. And then we have the data frame toy, and you see this would be that file which we have here.
Oprostite, profesore, da možete samo kako zove da ponovite kako ste taj toj ubacili gore?
Može, naravno. So we will get rid of this by like this. So okay, in order for you to upload any kind of text file or any data frame which you want to use in R, and we will need to use those because we will analyze and make plots out of them, you go here where it's written import datasets. Seems quite self-explanatory, right? And it is. And then we go from text base, right? Find it here. And then we find a file which we want to upload. Again, it should be a text file, which is either CSV, meaning comma delimited, so it's commas in between, so space delimited or tab delimited. It's all different files of text. But if you finish it with the extension TXT, you can assume it's tab delimited. If it's CSV, it is comma delimited, and so on. TSV is also a file type. I think it's semicolon delimited, and so on, right? So you find, and in my case, it's this genomics folder. I think you should have also those two files, and then you click here on the file, right? And then you see if your file has a heading, like a header above. In our case, our file did not have that. We didn't annotate anything. So we say that yes, make your heading, meaning that we do not have a heading, which is no. So it asks us, do we have a heading? We say no. So it puts you here. But if you say yes, it thinks your first line here is your heading, but it's not, right? So it would take this and make out of its header. And then you just click import. Now you have your file inside. You can repeat it as many times as you want. So you have, you can send import from Excel also. I didn't know that. In genomics, and we have toy pet, which is slightly different looking, but this is just a dummy file showing you how SNP data looks. right? And you can import it, and then you see here all these things, right? This is the base position, and it abbreviates it here a little bit differently, but the numbers are the same like in the file. Don't save. Okay, is this clear how you would upload data into this?
Yes.
That's good.
Yes.
That's good. So, uh, okay, we can come back now to. I'll do it like this. It's funny, I can't find my genomics file. Disk. It's weird. Okay, let's try like this. Good. I think this is the easiest way for you guys also to open your working directory, right? Instead of typing this like I would do, cd with this command, obviously would be good for you to know, but you hold shift, right? Hold shift, right click, and then open Linux shell here, right? And then you get this, and you're immediately in that folder. You can see we have here all this Plink and all this data here, right? Good. And now we can see, yep, for the girl who has a Mac computer, I think it's only one person. Could you just quickly share your screen with me? Is that okay?
Yeah, that's okay.
Okay. So I just want to see if it works, and then we can continue so you can follow and do the same thing as I do.
Oh. Okay, so which thing do you want to see exactly? The R Studio?
So no, now go to Plink first. Oh, sorry, go to Google, open Google.
I am on the Plink webpage.
Not this web page, just go to Google.
Okay. Okay.
Plink, just type Plink.
Okay.
Well, enter, right? So the first link there, click
Okay.
there. Yep.
Okay.
Download that one. Yep. Put it in the folder genomics, which you created hopefully.
Yes, I did. It's just on the side. Oops.
Oh, it's fine. Don't worry.
Sorry. I had made.
Just make a folder. Exactly right. So now, right, unzip it there.
Oh, okay. Okay.
Okay, open it. Now, good. So move all this stuff into the genomics and proteomics folder.
Okay.
Open the genomics and proteomics folder. Now open your PowerShell, like your shell script. You can try also by, I don't know how it goes on Mac, but you can also try holding Shift and then right clicking to see on the folder if it's open.
Which fold? This one?
Yeah, yes, go there, hold Shift and then right click or something.
This.
Okay, let me see. New terminal at folder, right? Go to the last one.
Okay. This.
One up.
This one.
Yeah. Click. Great. Now type ls. enter. Good. So now press dot slash and just type Plink. Press enter. I hate this with Macs.
I'm sorry.
But there's an option. Let me. Plink cannot be opened because Mac cannot verify. Okay, let me see. Cannot verify. Developer. Bypass. Cannot open user. Cannot be verified. Okay. Quick steps. Right click on the app and select open. Click open again. Click on privacy. It's so complicated for no reason. Allow apps download from anywhere. Remove quarantine. Remove. Okay. Go to the. I need to look at your screen now. Cancel that. And now right click on Plink. On that Plink.
This?
Yep. Yes. Get info. Try get info. Okay. More info. Comments open with preview sharing. Sharing and permissions go down. Doesn't work right.
No.
Okay, maybe you can go like this. So go open system setting preferences. Just type it in that system setting.
Like here?
Yeah, try to find it. You can go privacy and security. Yeah, I see it down on the left side. Just X that part.
Oh,
Go out the other window, the window below.
oh, this one. Okay.
And now go to privacy and security. A little bit down, a little bit down, a little bit down. Yeah, click that.
Okay.
Okay, let me see what this says here. Click open anyway. What the fuck? I hate this. I can't tell you how much I hate my computers. I really do hate the whole firm, and I hate Tesla too. I hate those both particularly. They make stuff complicated for no reason.
Same.
Well, maybe someone had that question for Blink. I need to fix this now, so later you will never do it by yourself, I guess. Open an app map from under. Then choose Launchpad to control the app icon and choose open from shortcut and click open. Okay. In the Finder on your Mac, locate the app you want to open. Don't use Launchpad to do this. Launchpad doesn't allow you to access the shortcut menu. Control click the app icon, then choose. Okay, try control click the app icon, whatever that means. Hold control and then click on it, I guess.
Which app?
The Plink. Plink is an app, right?
This?
Yeah.
Okay, control and this.
And what says afterwards? Click control, then choose open from shortcut menu.
Huh?
No. Try open with terminal, maybe?
It's the default.
Yeah, just go open. Yes.
Okay.
Yes. Good. Now you see you can scroll up. You see now it opens like all the stuff from the software. Like the commands include make, bad, blah, blah, blah. So that's good.
Okay.
Okay, and now try to click something. Try write something.
Here?
Yeah.
I can't write
Okay. hold Control and press C. Try just clicking escape maybe.
I did.
Doesn't work.
No.
Just X that window.
This?
Yes.
Okay.
And try now to rerun this again on this. No, just on this other window. Yeah, there.
How? What do I do?
Just click your arrow up.
Okay. And see?
No, just your arrow on, the arrows on your keyboard. Just the arrow which shows up. Strela gore.
Yeah, but to a control.
No, not control. It's just,
Like this?
yeah.
Ah, okay. Okay.
Now enter.
Okay.
Yes, it works.
Okay.
Cool. Good. Don't have to share the screen again.
Okay, thank you.
Whew. Okay.
Sorry.
We fixed that. So yeah. So now we'll continue by. Sorry. Now we'll continue by looking into data and how data Plink actually does data analysis. Because we will not be able to generate our own data files for some time, but not to, because we need to go through the lectures first, right? To understand what's going on with the sequencing and so back and so forth. But nevertheless, we will be able to look into dummy data, which is already generated, and this is the folder which you have when you extract Plink. You get those toy map and ped files, right? And hopefully you have some text editor. In my case, this is Notepad++, which for some reason now doesn't respond. And I'll just show you how, yeah, good.
Can you share your screen?
Is that out? Not share, sorry.
Yeah.
I thought I'm showing you the whole time. So this, for example, is a raw file, right? We talked about this last time. This is just ATCs and Gs, raw file, nothing done to it. But once you get all the different snips and all this different stuff going, you technically get either a VCF file. We talked about there's several file types, but also you can get very simple files like map and ped file. And map and ped file are so-called binary files, which even though they're two files, this is actually one file, right? Because let's explain it maybe like this. So this raw file, which we have from the sequencing machine, takes some time to open, right? We do all the QC after we know what QC is going on, blah, blah, blah. And then in the end, we get either a file of this caliber or a file of 1, which is a VCF, or a file of 3, which is a binary or not binary file. It's called binary, but it's 3 of them, which is BIM, BED, and FAM file, also denotating the same thing, right? So imagine once you do the sequencing analysis, you can get either one file or two files or three files, or representing the same data, right? And the reason is quite simple. If you have only one file, it takes a lot of time for the computer to go through all the lines in the code, because it could be several thousand or millions of lines, right? But this is good if you want to have additional data in this file, like pathogenicity of a variant, or I don't know, any kind of additional comments you would like to have in the file. But that is most of the time a little bit too much, and you don't need this many information in a file, so you split the data from the file in two files, right? Which would be then a map and ped file. And it's very simple. The first place here in the ped file will tell you which chromosome it is. Here, then you would have the chromosomal position, or the base pair position, rather, on chromosome 1. And here you would have a denotation of if the patient is a case or control, if the patients are in any relationship to each other, if they're a family, and then if they're male or female, for example. And then we would see what they have as a genotype for this particular SNP. So imagine this line of this file goes with this line in this file. So basically here we have the RS ID, so the SNP, and then also where the SNP is, and it tells us for this SNP we have a 0, 0 genotype, meaning that this SNP was not successfully genotyped for this patient, for example, right? And the other one, RS10, for example, this would be the position here, AA. So for this SNP, this patient would be AA, or this person, meaning that for this here, the other one would be CC, and he would be AG for the other SNP. So it's splitting data into two files for the computer to be able to easier analyze and to do it quicker. And there's even a third option where you have 3 files, which we will show maybe next time, where you have similar splitting of data plus a cartography file called a bed file, which is basically something you cannot open on your computer. It will show you some gibberish or some hieroglyphics, basically, where you'll not be able to read anything out of it, but it also uses this to quicker access the data. So once we'll see when you type the commands in Blink, I want to extract this SNP and this SNP. In order for it to go, the software, like line by line by line, to find your particular SNP, it uses this cartography file basically to be able to find the SNP faster, right? And in order for us to really grasp all this stuff, I would suggest you to go to this web page, and I think I mentioned it already, where you can actually have a look in how this data looks. And why is it important to use all of those commands here in order to set up, you see, binary PET files. This is what we have here. And I think we can maybe try to see if it works with this one. So we go Clink, and we use the command file. We use our toy file, and we want to say make out of it also a toy file, but it will be called toy1. Okay, and now it said something that it has done something here, right? And we go into our folder and we see 3 new files. We had map and ped, but now, as I mentioned, we have the bed, bim, and fam files. And now we can compare and open those to each other. We need to do like this so you can see better. So we take the fam file, we take the bim file, and we take the bed file. And you can see the bed files write something which we have no idea what it is, some odd numbers and letters which has nothing to do with anything. The BIM file looks kind of similar to this in the PED file, and the FAM file looks also similar to the PED file, but also in combination, sorry, map and this and this and this look similar. So it's just, the data is the same, it's SNPs, it's patients, it's positions, but it just differently shows the data points for the machine to be able to analyze it. And there's another way of looking into this, where is instead of make bed would be a command to make out of this binary file this other file, this 3 kind of structured file, and I think the command VCF will make us a VCF file. Did it? Maybe it's recode VCF. Okay. Now we have a VCF file too, which is only one file, which then looks like this, right? All those three stuffs are the same thing. Identical, same raw data behind it. It's just transformation in order to represent it differently. And if you banalize it even to the core even more, this is just text files. You can make a genetic file by yourself, right? So, and how would I do this? Enter. I use tab because it's tab delimited. I type I'm just making this data up. I call it 2023. Do like this and do like this. Now I have 3 SNPs here, and I do this, and I do this. Sorry. You should now have 3 SNPs inside, right? Because in the end, it's just a text file. Nothing more, there's nothing spectacular to it. And technically you could, once you figure out here in the text how this file is structured, you can just go and play around and go open a text editor and make 2 files, and one called save like test.map and test.ped, for example, and then you can upload it like I did here, right? Again, it's all only text files. You only need to know the structure of it in order for you to be understanding on how to manipulate it and how to get data out of it. And this is basically, I think, enough for today in regards of setting up software. Does anybody have any questions in addition to this?
All clear.
Just a second. Samo sekundu, hoće da mi šlepaju auto. Ja se izvinjavam. Moram točno pa će mi auto. Svašta. Dobro, do you have any questions?
All clear.
No, everything is clear.
Okay, good. See you then next week. Bye.
Goodbye.
Profesore,
Molim.
imam ja jedno pitanje. Samo.
Ja.
Ove prezentacije, one trebaju biti sve u srijedu, jel tako?
Ja.
Ja imam mali problem, ne znam da li možete izaći u susret. Imam let u srijedu u 4:30 i nikako ne mogu da stignem na to, na te prezentacije. Ne znam da li slučajno može moja grupa neki drugi dan da prezentuje.
Može, ajde čućemo se, ajde, moram ići, čao.
Hvala.